I'd like to welcome everyone to our first session this morning. The topic of our morning session here is peering inside cells to understand subcellular compartmentalization and ultrastructure. So we've got a number of really interesting talks lined up here. So without any more delay, we'll go ahead and get started with our first speaker. Elizabeth, if you'd like to start sharing your screen, I can go ahead and introduce you. So our first speaker is Dr. Elizabeth Wright. She is a professor at the University of Wisconsin-Madison College of Agricultural and Life Sciences. Dr. Wright's research uses cryo-EM to aid in the development of novel antimicrobials, therapeutics, and vaccines. Her talk this morning will cover recent developments in whole cell cryoelectron tomography. And it looks like we can see your slides. Can we hear you?
Yes, thanks.
Yes, we can hear you. All right, go ahead.
Great. Thank you, Trevor, for that great introduction, and thank you to all the organizers for inviting me to speak in the EMSL Integration 2020 meeting. So as Trevor said, I'll talk to you a little bit about what we've been working on to develop whole cell cryo-ET for studies of, in our case, we're very interested in host pathogen interactions. So in my laboratory, we work on a lot of different research projects. The thing that ties us together is really how we're developing the technologies to advance our understanding of the different biologies we're focused on. And so we do a lot of work in developing correlative microscopy, so bridging between fluorescence light microscopy and cryo-EM. We've been working on phase plates for a number of years to enhance contrast in the electron microscope. And we also use affinity capture methods to help us adhere particles of interest to grids. And so with using these technologies, we are investigating a lot of different structures in bacteria, and in the case of today's talk, looking at envelope viruses and their structures. So when many of us think about the future of structural cell biology, we're working to develop tools that help us move beyond just imaging in isolated sets of experiments, but working to bridge across multiple disciplines. So if we're developing tools to better understand molecular and physiological data on the bench, we can really relate that to what we're seeing in the microscope, because each one of these target areas has a particular strength in giving us information about the sample we're studying. But when they're in isolation from one another, it's very hard to correlate between them. And so our goal with many of the techniques that we're developing in the laboratory is how can we develop substrates or smart surfaces that we can then probe with molecular and physiological experiments, and then do imaging at different modalities on those same samples to begin to investigate the ultrastructure and the high resolution structure of different macromolecular complexes within them. And so an example I can give is, since we work on a lot of viruses, and we would say, look at this fluorescence image in the middle, and the little red squigglies are RSV virus particles. Everyone asked me, well, which one is the infectious virus? And so I can give a global estimate based on titration data and infectious particles that we can purify out. But to be able to actually pinpoint to a particular virus particle and say that is an infectious agent is really key and critical to our better understanding of these complex systems. So a brief outline of my talk today. I'll talk about some of the technologies that we've been using and implementing in the lab for preparing envelope viruses for cryo-EM, how we use whole cell cryo-ET to investigate a respiratory syncytial virus vaccine candidate, and then how we have also been using whole cell cryo-ET to further define virus assembly. So what is respiratory syncytial virus? There's a lot of news out today about different viruses in the world. Measles virus is one we study. Coronavirus is on everyone's mind right now, but RSV is a particular challenge for the pediatric and also the geriatric population. It's a pneumovirus, it's an enveloped virus, and it impacts the respiratory system. So it can give us bronchiolitis, pneumonia, and many times children who are infected when they're very young can lead to the development of asthma. At this point in time, there's no vaccine available. So many groups are working hard to develop one. There is a monoclonal antibody treatment, but this is only available to infants at high risk. So commonly, if your child or infant is infected with RSV, the doctor will say, keep them comfortable, rest, fluids, in hopes that the infection will pass. So our focus is to really work to understand the structure of these viruses so that additional targeted therapeutics can be developed, and also that we can work with groups to develop vaccines. So briefly, I'll give you a little idea of what RSV looks like. So the genome structure is up top. This is an enveloped virus, so it buds from the cell membrane. And so this is where the gray line is on this particular cartoon, extending from the outside of the virus. In green, we have the F glycoprotein. We also have in magenta, a small hydrophobic protein, and in blue, the G glycoprotein. And so these, in concert or in isolation, regulate the virus's attachment to the host cell. Underneath the membrane, we have the matrix protein, and then we have the M21 protein. And then within the interior, we have the RNP complex that contains the ribonuclear protein, or the genome, that's the RNA genome, encapsulated in nucleoprotein, and P and L are in conjunction. And so our interest in studying RSV is really to determine the native state structure of this virus, understand its assembly process, and how it interacts with host cell. And so why are these viruses tricky to study? Well, in a nutshell, they're enveloped viruses. So what does that mean? These viruses derive their outer coat, if you will, or their envelope from the host cell or a host cell membrane. And so many times these viruses are budding from the host cell, in the case of RSV, HIV, flu, and measles virus. And so when we look at these viruses, because they have this really flexible, dynamic outer membrane coat, they can be very fragile when we're imaging them in the microscope. They can also be extremely pleomorphic, which introduces challenges when we want to use cryo-EM techniques to study their structure. And so when we think about what's the appropriate method to use to study RSV or these other envelope viruses, we really focus on using electron tomography, because each one of these viruses is pleomorphic. So while if we look at them in a broad scope, we can understand that they have the same structural proteins, genome is similar, et cetera, but each virus particle, like a human, is unique. And so we use electron tomography to capture the images in a tilt series of the viruses as we tilt them incrementally in the microscope. And so that's what this diagram is showing, that the electron beam is passing through the sample. We take a tilt series, and at particular degree tilts, we collect those images, and then through a process known as weighted back projection, we generate the 3D reconstruction of that individual particle, and then thousands of different particles or viruses that we're interested in studying. So with many of the things we look at by EM, we think of how do we prepare them for imaging them in the microscope. And in the case of proteins, macromolecular complexes, viruses, even parts of cells, we typically do some form of purification. But in our lab, we've been beginning to wonder, is this a best practice that we can use for EM imaging or any imaging that we want to do to understand the structure of these particular particles? And so in the case of these envelope viruses, many times we start with a cell culture where we've infected them with viruses, we watch the viruses propagate over time, and then we pour off or scrape the cells, we pour off the supernatant, we do low speed clarification to get rid of cell debris. Many times we do harsh treatments, downsing, sonication, freeze-thaw cycles, and remember, these are envelope viruses, so they're really just bound by this very fragile lipid membrane coat. And each one of these steps, because it's an artificial step that these viruses, or if you're looking at cells of any sort, they're not used to going through. And so these are very artificial methods. So these can damage the particle at the front end. But one of the other challenges we have with many of these envelope viruses is they grow to extremely low titers. So typically, it's a good day if we get a viral titer of 10 to the 7th particles, but that's very few particles when we spread them out on an EM grid. We can have challenges with impurities, so cell debris still being present, and of course, damaging the virus particles. So how do we think about resolving some of these challenges with doing harsh purification? Well, if we're going to look at them at the first, maybe we have no choice. And so we're going to look at purified virus particles. And we can see these preparations of HIV, flu, and Newcastle disease virus. And so these are pretty good. We can see the virus particles present on the grid, in various distributions based on the number of virus particles in the preparation. In some cases, there's very few. In some cases, there's many. But then you can also see that their distribution across the grid can be very random. So in some cases, you'll have the high dense packing in the case of the flu virus, or they're clustered, or you only have one particle in the case of the Newcastle disease virus. And so ideally, we would want to overcome a number of these challenges through various means of preparation. So as I alluded to, there's poor particle distribution, uneven particle distribution. We have this low titer problem and damaged particles. Well, what are our solutions? We could work on different systems, and in our case, we could work on bacteria as part of the lab does, but the virologist in the lab wouldn't be very happy. We can figure out how to improve our purification schemes. Is that possible with some of the viruses that we're looking at? We could stop relying on purification, or we could think about how we can actually selectively capture the objects on the grid that we're interested in. And so just as a highlight for RSV, we worked very hard to purify virus particles of RSV, but again, we see, a wide range of morphologies of these virus particles, and what does this mean to us? In a nutshell, when a virologist looks at these with us and we're showing them, hey, look at what RSV looks like, they typically ask, well, which one represents the infectious virus? Now, in many cases, we have prior knowledge that tells us that one trajectory is correct, but if we're looking at a new virus system, this may not be helpful. So as I mentioned, one of the techniques that we think about using is affinity purification. So can we use tools such as those that we use for column purification of proteins to be able to selectively capture viruses on grids? And so this was some work that Debbie Kelly did a number of years ago to develop affinity capture techniques for EM grids. And many people now have developed extensions off of this using different substrates and linker agents to target particular macromolecular complexes. But this is really a simple, elegant technique that anyone can do in the lab. So you basically lay down a lipid monolayer on your grid surface. Within that lipid monolayer, you have your inactive lipids, and then you have your active lipids that have nickel NTAs associated with them. And so you can directly interact a his-tagged protein, onto that nickel NTA. In the case of maybe you don't have that, you can use a his-tagged protein A that links to the nickel NTA, but then combine to your antibody and then your target protein of interest. And so the end goal of using this technique is to go from a grid surface where you have all kinds of different complexes or virus particles and debris spread across the grid to where you can have a very uniform, a more uniform distribution of those virus particles with very little trash in the background. In some cases, if you're looking at smaller complexes, this can limit preferred particle orientation. This can depend on the linker complex that you're using. You can also, in some cases, minimize challenges associated with the air-water interface, because this can also lead to incorrect understanding of a structure that you're interested in studying. And so what did this allow us to see in the case of measles virus, another envelope virus that we study? Again, it is present at very low titers and preparations. So if we're looking at an untreated grid, we see all these particles on it and we think, wow, we've got a great number of virus particles on this grid. This is really exciting. But when we actually go in and collect tomography data on this, we see maybe there's one virus particle in a field of cellular debris. In this case, we used a 20% nickel NTA, and you can see this really cleaned up the grid quite a lot because we were interacting with complexes on that virus particle, i.e. the glycoproteins on the surface. And so we were tethering against them. And you can see now that only particles that had the glycoproteins on the surface were interacting with the nickel NTAs on the grid. So it's really dramatically reduced cellular debris. This worked for measles virus, RSV, HIV, Newcastle disease virus, and it really improved the measles virus distribution on the grid so that we could have more particles that were measles virus and more of them would be present in the holes where we're ideally collecting data. But again, I should mention that these were actually purified virus preparations. And so when we were studying these virus particles, not only our group, but Sarah Butcher's lab was looking at measles virus at the same time, we were resolving some challenges that were unanticipated with the structure of the virus. So if you look at the top two panels, especially where this black arrow is pointing, when we study the ribonuclear protein complex and look at it in isolation or in many virus particles, it should be this single herringbone-like structure. In this case, we were seeing a doubled structure where the RNP was coated with another structure. And so Sarah's group did some really beautiful immuno-EM and illustrated that this was the matrix protein. And so in essence, what was happening is for some reason, the matrix protein was coating the RNP, which is very unexpected when we look at paramyxo or pneumoviruses. And so we wondered if this was an alternative assembly pathway for these viruses that may be replicated amongst other viruses in these particular virus groups. So this is a challenge when we do purification, that it may lead us to thinking of hypotheses that are not factual. So are these assembly pathways relevant, i.e. is the assembly pathway where the matrix protein coats the RNP complex the true viable infectious virus particle? Or do we think of the more canonical model where the matrix protein is coating underneath the lipid monolayer as what's actually happening with these viruses? And so our only experimental solution was to develop whole cell cryo-ET so that we could tease out what was the viable way that these viruses assemble and replicate and then infect the next host cell versus something that may be a disrupted structure. And so this process is pretty simple. We can do any type of cell that you're interested in as long as we can kind of adhere it to a grid surface. So we've used many standard cells, primary cells, bioproduction cells. This has a great level of scalability, so we can do multimodal, multi-scale imaging workflows. And we can label any complex within the cell or the virus so that we can really have a broad correlative imaging pipeline moving through this process. And so this image up in the upper right-hand corner is showing you of some cells growing on a grid. This is a relatively good distribution. You don't want too many per grid square or else your ice is going to be too thick and maybe you'll have some artifact within that ice. And we're just showing here the difference at the bottom, the different cell types that we use in our laboratory, but we also use neurons, macrophages, look at eosinophils, lots and lots of different cells. As long as we can adhere them to a grid and help them spread out, we can image them. The Z slices are just showing you the different thicknesses that these cells can be growing at on the grids. And so in some cases, like HeLa cells are kind of the perfect cell because they can spread pretty thinly. And so there's a lot of area that we can image through. Whereas some of these other more elegant cells, like these Niemann-Pick cells, they're a bit thicker. They're a cholesterol kind of modulated cell line. And so we just have to be wary about how we're using them in our experiments. And so here's a little bit about the workflow. In this case, when we're studying respiratory syncytial virus, we conveniently have this integrated fluorescent protein tag, MCATE. So it's a Katushka tag that's located at the 3' end of the genome. And so this allows us to see which cells on the grid are replication competent for the virus. And then to further understand where the viruses are assembling on these cells, we can native immunolabel the RSV-F fusion glycoprotein on the surface. So here, Jay developed the immunolabeling system where you can see the different cells. She's grown them on the grid, seeded them at sufficient levels, examined them by fluorescence microscopy, then she's plunge frozen them. We have multiple different plunge freezers we use. Depends really on the cell system that we're investigating, which one works best, because cells, especially mammalian cells, are very delicate. So if you're blotting them with filter paper, you have the high likelihood that you're going to rip off part of the cell. So maybe the better method is to blot from the backside, so you have less likelihood of damaging the cell. Then we can go in and do cryofluorescence microscopy to pinpoint and target our areas of interest that we're going to then collect cryo-ET data on. And so with using these techniques, this is what the native particle morphology for RSV looks like. And we can see that in here we have the HeLa cell body at the bottom, at the top panel. And so this is a pretty thick region of the cell, so we're not going to be able to have the electron beam penetrate that, but you can see all the virus particles growing off of that cell. And in the case of RSV, we really found that about 94% of the virus particles from multiple different virus strains, multiple different cell types, were filamentous. So this really gave us great confidence that the virus particle that our body is going to see in an active infection is a filamentous virus particle. Here is just a brief movie illustrating the value of using cryo-ET to investigate a whole cell in the process of virus particle assembling. And so we can segment these structures that are of interest inside the cell to help us understand what's at a site of virus assembly. So the microtubules, the actin network, ribosomes, and many, many other structures that would be present, as well as all the viral protein structures that have been recruited and assembled to those assembly sites. So what does this mean for vaccine design? I've shown you a little bit about how we can understand what the virus looks like, in its regular context, but what about if we really want to develop a vaccine? And this can be useful for not only vaccine candidates for viruses, but bacteria and other entities. And so early on, RSV vaccines were developed in the late 1960s, but the challenge at that point in time was they were using a formalin inactivated virus. And so when they introduced that vaccine candidate to a number of children, it made them very sick, and also several deaths resulted. And so vaccine development in the field of RSV research basically stopped for a very, very long time. And people were working to develop these monoclonal antibodies and other therapeutics to support those that may be infected. And so it wasn't until recently that people began to think about, well, what if we develop live attenuated RSV vaccines? Because you can balance the attenuation and immunogenicity, you can modify them genetically to be unstable, but could this cause some physical instability? And so one of our collaborators, Marty Moore, who was at Emory, now has his own company, developed a codon deoptimization methodology to make a live attenuated RSV vaccine. And so what does that mean? So with The codon deoptimization, we normally think of that we want to optimize the codons that are present when we're generating a protein. Here, what Marty did was he modulated them to down produce particular targets. So in this case, he codon deoptimized G. And so G is the attachment glycoprotein. Now G is present on the virus surface, but is also present just in suspensions of the virus. So it's a released complex. And so by decodon optimizing G, what he hoped to do was reduce the amount of G on the surface of the particles, as well as what would be present in loose form. And so when we collected data on this vaccine candidate, what we found was, now we're slicing through the 3D volume, so we're now at the interior of the virus. But what's most interesting is when we look at the surface. Maybe I'll pause here. And so if we look at the glycoproteins on the surface, they really organize themselves in these hexagonal arrays. So it was hexagonal packing of either F or F and G on the surface of these viruses. Now we've looked at RSV for a very long time, and we've never seen this type of packing before. And so this really indicated to us that there was an alteration in the structure and organization of the glycoproteins based on the modulation that Marty had done. And so to examine this further, to begin to understand what the F functional state was on the vaccine candidate virus, we needed to do a bit more back of the envelope work. And so here we can actually use heat treatment to inactivate a virus. And so what this will do is it'll trigger conformational changes in the viral proteins. For example, RSV-F, i.e. the glycoprotein on the surface of the virus, the fusion glycoprotein, and the matrix protein, which is underlying the membrane. And so you can see this in these viruses here, where we very dramatically changed the structure of the particles, moving from a filamentous particle to a spherical particle. And so we can do this, so you can see F looks very different here. On the spherical particle, it has this more triangular shape, whereas on the non-triggered particle, it's more in the pre-fusion conformational state. And so we did a number of measurements to assess the ratio of F in the pre or post-fusion, i.e. triggered conformational state on these virus particles to confidently assess their structure. But then we could apply to the virus particles that we were most interested in, i.e. the vaccine candidate, to validate which structural state that glycoprotein is in. And so we can best do this with a technique added on computationally to cryo-ET known as subtomogram averaging. And so what we do here is we take a tomogram, we cut out 3D volumes of that tomogram, and the structures within them are going to be all randomly oriented. We align them to a reference, as we see here. And then we iterate through this process of averaging all these tomograms or subtomograms together, maybe extracting some that are not good until we get a higher signal to noise and potentially higher resolution structure of the object of interest. And so this is what Zenlong did for these virus particles that we were interested in, where we could see that on the A2, which is basically the lab adapted strain of RSV, OE4, which is the vaccine candidate, the largest population of the glycoproteins on the surface of those were in the pre-fusion conformational state, i.e. an inactivated, a form of the virus fusion glycoprotein that has not interacted or responded to a triggering event. Whereas if we look at the heated glycoprotein, it is triggered into this post-fusion conformational state. We could also examine this with native immunolabeling, either in cryo-ET form, as in the top panels, or with thin section EM on the bottom, if we wanted to quantify in both states. And so we can then pinpoint and calculate the ratio of the number of pre-post fusion F glycoproteins and the ratio of G on these virus particles, and assess if what we had done genetically had modulated the populations on them. And so that's what we did here. And on the OE4 virus, we can clearly exhibit a greater density of pre-fusion F than on A2. And there was really no difference in the amount of post-fusion F, which is a good sign that we had actually modulated to retain pre-fusion on the virus particles genetically, as well as the all the manipulations we had done to understand the structure of these particles also did not inadvertently trigger F to the post-fusion form. We also found that the G protein density on the particles was approximately half of A2. And this is really through the codon deoptimization process. So with all the viruses that I've shown you, I've talked very little about the matrix protein. And so this has been an exciting story for us, is to really understand what this protein that lies at the interface of the membrane of the virus, just under the membrane, what it's doing to regulate the structure of these virus particles. And so it's a small 28 kilodalton monomer. It works as a functional dimer, as you can see here. And it interacts with F and G on the surface of the virus particles and M21 on the interior. And so this is what this density plot is illustrating, is that plot, peak 1 is the F glycoprotein, 2 is the lipid membrane, 3 is the matrix protein just along the interior of the lipid bilayer, 4 is M21, and 5 and 6 are the ribonucleoprotein complex. Now, in all the data that we've studied up until now, we've seen the density of the matrix protein, but we haven't really been able to understand its structure to any great detail. And so recently we were able to collect some data at PNCC, and I've highlighted this area in the blue box so that you can kind of orient your eye when we go through, slice through the volume of these virus particles. And I hope as we slice inside, so just glancing, that you'll actually see this crosshatch lattice just under the lipid bilayer. And so this was the first time we saw the matrix protein lattice on these virus particles, and we've really imaged thousands of particles. And it was with using the great resources at PNCC that allowed us to see this at last. And so much like we did with the F glycoprotein, but in a slightly different way, because we're really interested in understanding the organization of this matrix protein lattice, we did subvolume averaging. So Brian initially picked a random organization of particles to get a baseline model of what this lattice would look like. And then he reintroduced to force Pete, which is the program he was using, to automatically pick positions where there would be matrix protein lattice so that we could generate a higher signal to noise, higher resolution structure of this particular lattice. So the density is illustrated in the dark on this. And so what we found was the spacing of the matrix protein lattice is 6.2 nanometers roughly in one dimension and 5.2 in the other, which is really is in great agreement with the crystal structure lattice of the M dimer. And we also found that the lattice also has a helical orientation, which is at a roughly 45-degree angle. And so this makes sense because these viruses are filamentous and they're forming as a filament. So you're expecting this lattice to curve around the filament as it extends outward from the cell as it's growing. So I hope what I've showed you today is that we can really think broadly about how we develop and apply methods in cryo-EM to optimize many different studies of envelope viruses, but also mammalian cells and bacteria so that we can minimize destruction. The RSV is a filament versus a sphere. We can use these techniques for vaccine design. And that finally, we've resolved the matrix protein lattice with using these new tools. So I see Trevor there. I'll highlight a few people real quick. We've recently moved to UW-Madison. So Jay, Brian, and Joe have been the ones to really push the envelope and move forward with the whole cell cryo-ET work, CLEM, and the viruses. We're grateful to the resources at PNCC, where we collected the most recent data, the whole host of people that worked with us at Emory, funding sources. And of course, I have to give a brief plug. We've just opened up our new Crylium Research Center at UW-Madison. We're looking for a great number of people. And we've just recently been funded to be one of the national NIH National Center for Cryo-ET. So we'll be the hub. And so this is the Midwest Center for Cryo-ET, which will really support the whole research environment where you could come and get access and training to cryo-ET specimen preparation, data collection, and computation. So with that, thanks, and I'm happy to answer any questions.